presentation. It's a huge honor as an electrical engineer to get the chance to come full circle and get a chance to speak with some people who are much more rooted in the roots from which I come than the biomedical engineering territory in which I'm forging my new path. Okay, so, this is me. Hello, that's my email, I suppose. How are we doing this? All right, so, next slide, please. I guess this isn't radio controlled. Okay, I'll use hard keys. Hi, Ross. I'm hitting next and I'm having a problem here. Oh, well, let's figure this out. All right, so everyone, meet Noah. When he was 10 years old, he was playing in a tree in Central Park and he accidentally fell out of it, breaking his ankle, as you can see. How American, right? This American flag cast. And he broke his ankle, unfortunately, in the growth plate, which, for those of you who don't know what this means, meant that his left leg was growing slower than his right leg. Okay? So over the course of the next 20 years, he had revision surgeries, several of them. This is the only surgery picture I'll show you. Ouch! Recovery. And I won't show you where he is now. I met him surfing in Costa Rica. He's actually my fiance. We've recently gotten engaged. And he's doing fine. Okay? But the way that they repaired his ankle is actually quite important to know, because they took a piece out of his hip bone. He calls it his shark bite. And they moved it to his ankle. Okay, so it worked. It worked really well. But the thing is that for so many surgeries like this, whether it be for cancer, or for trauma, or congenital defects, or whatever the reason, there are over 2 million of these procedures worldwide every year. The only way, even now, to get human bone, when you need it for any of these procedures, is quite literally to cut it out of a human. And when you consider that we have stem cells living in our bodies that repair our bodies every day, it seems like a quite brutalist approach. And so when I think about where we are now, we're actually at the cusp of a revolution in how we view the body. I like to call this body 1.0, 2.0, and 3.0. You know, up through most of human history, we viewed the body as a kind of intact unit, like Leonardo's Vitruvian Man, right? I love this image. And implicit in this idea is that if something was wrong with the human body, if you just let it go, let it right itself, it would come back to rest. But somewhere around the turn of the last century, as we started developing things like interchangeable parts on the assembly line, we started to view the body in a similar way. If you needed a new heart, you might get one from a donor, or you might get, engineer an artificial heart in the lab. But the idea was still the same. We were like C3PO, right? A machine. And therefore, if we were to be, repairing the human body, we would take the view of a mechanic. But actually, as Patrizia mentioned, we have trillions of cells living in our body. What she didn't mention is that we actually have 10 times more bacterial cells than human cells in our living body. Okay? So we are a veritable ecosystem of cells. 10 times of us aren't even human. And yet, these cells almost conspire in a way, despite what we do, in order to keep us alive. And this is what I think is really interesting, especially from a data science perspective. You might be wondering, who am I? I'm dreading this next slide. Yeah, so we all have a picture like this, right? You're 8 years old, you get your first chemistry set, right? Your smile's as big as your hair. So this is my family. These are my siblings, Sheila, Mira, and me, and my brother Neil, and we're playing in a science museum in Toronto. And when I look at this picture, I could go in many directions, hair notwithstanding, and I think to myself, this was a time in my life when we received a pretty sobering diagnosis. My 2 sisters, Sheila and Mira, were diagnosed with retinitis pigmentosa. This is an X-linked degeneration of the retina condition. My brother Neil, retinitis pigmentosa for my brother Neil, and color blindness for my 2 sisters. And all of these are carried on the X gene. And what this meant for us as kids is that we discovered that we all saw the world in this really kooky and interesting way. But it also meant that it was rooted back to our genealogy. And this was a really fascinating moment for me as a kid, because I realized, like many of us do, and our next speaker will really talk much more about, is that our perceptions are based in ourselves. And that's pretty amazing. It's miraculous. But it's also fallible. It's basically like any other technology, isn't it? And since you guys are mostly an IT crowd, I guess I can sort of nerd out a little bit and say how I follow this professional preoccupation through a number of turns, including electronic musical instruments. This is a theremin for any analog music nerds out there, through computer vision, and a stint in telecom, programming voice over IP networks, until I landed in a field called tissue engineering at MIT. And this is a piece of cardiac tissue that I engineered during my PhD. As you can see, it's beating. It's beating actually in time with an electronic circuit that I programmed using some thick microcontrollers. This was a while ago. And it's beating in time with this circuit. And for me, this was a really interesting thing, because I could look into the microscope, and it's one thing to build a circuit that you plug into the wall. It's another thing to see it interacting in real time with something that's alive. And this was totally fascinating for me. I remember looking down into the microscope and seeing these cells beating and just feeling so honored to get a chance to be doing that research. But I also felt that it was, the Bush years, there was a lot going on in stem cells and controversy around that. And I realized that at the same time that I was looking into this microscope and seeing these cells, I was also inextricably linked with what was happening in the political sphere and in the business sphere. So actually, after my PhD, I left academia to join McKinsey and did a lot of work studying pharmaceuticals and medical device strategy for people launching big products. And of course, the grass is always greener when you work somewhere, and so I missed the pipette as I was working in PowerPoint and came back to the lab where, as Patrizia mentioned, I co-founded a company after I came back to academia called EpiBone. So EpiBone represents a kind of U-turn back into academia and then a launching point into the startup sphere. And so we're using this principle of viewing the body as an ecosystem, okay? So what we're doing with EpiBone is we're generating living, personalized bone grafts that are correct, not just from a 3D fabrication point of view, but also from an immunological point of view. So we're engineering living implants that can hopefully serve the needs of people like Noah and others. So many people don't know this, but after blood, bone is the most transplanted material in the body, amounting to millions of procedures costing billions of dollars worldwide. And as I mentioned, we're still just cutting bones out of humans to get these tissues. But the way that we do it with EpiBone is a different approach. We're viewing these cells that are in our bodies as living building blocks. And so we take an image, okay, this is just 3D data. We take a CT scan, right? AKA a three-dimensional X-ray, and we extract the three-dimensional data out of that image in order to generate X code for things like 3D fabrication, like micromilling machines and 3D printers. And we engineer a perfect 3D puzzle piece that fits the defect, okay? Then we take stem cells from the patient, so we take a small tissue sample of adipose tissue, and we extract the stem cells out of that. That's just a polite way of saying fat tissue. We extract the stem cells out of that and infuse that into this customized scaffold shape, and we mature those cells in that biomaterial for 3 weeks. And then we have a piece of living graft that's ready for human implantation. Here's a short video explaining the process. So these are the cells. We're machining the graft, growing up the cells in an incubator. That's our bioreactor, feeding the cells over time, and voila. After 3 weeks, a piece of living tissue that's ready for implantation. There are many benefits to this approach. Firstly, because there's a perfect fit, there's no need for surgeons to machine grafts in the operating room. Secondly, because it's your own cells, there's no mismatch of the code between what the implanted tissue is and what your own body expects. And thirdly, because it's alive, we hope that it will continue to remodel and grow with the human body. We've gotten a lot of press to date. It's been a really fun journey so far. It's been about 18 months since we fired ourselves from our academic appointments and hired ourselves at the startup. And we've raised a total of about $6 million from several different sources if anyone's interested in learning more about academic entrepreneurship. And even though, so now we're based in Brooklyn, we're 8 employees, and we've been designated World Economic Forum Tech Pioneers, which has been an amazing boon for us as we think about our global strategy. But I was given permission rather, to be a little bit provocative today. And so what I hope to really spend, what I'd really like to be able to touch on in this conversation is this idea. You know, a lot of people ask, you know, how do you devote yourself to this process that's going to take 10 years or longer to carry out? And for me, you know, a lot of times I think, if there were a movie that I'd love to be an extra in, what would that be? And I think for me, this story of being at the cusp of a new view of how we envision the body and what that means about new therapies going forward, as well as other technologies that might intersect a little bit more with the world that our audience lives in. What I think is that there's a paradigm shifting question that we can ask ourselves that's kind of implicit in this story. You know, if I think about these cells that contain, you know, gigabytes of data each, of which we have trillions of in our bodies, and are essentially living factories that produce lots of proteins every day in and day out, billions of cells, trillions of cells. In the case of the heart, billions of beats in a lifetime, never pausing to rest. I mean, these factories are amazing. And what would happen for a wide range of industries if we started viewing them as living building blocks, okay? Analogous to our transistor, which, as we know, we've gotten a lot of mileage out of. And so I showed you all an embarrassing picture of me, so I figure I'll show you all an embarrassing picture of you. Because after all, we all looked like this at one point, didn't we? We were all 1 cell big, 2 cells big, 4 cells big, 8 cells big. You know, you guys know binary, right? You can do the math. But how did we get from being 1 cell to the trillions of cells that we are, each of us? And not just human cells, bacterial cells. How did we become that ecosystem? And of course, that's the realm of developmental biology. Understanding those biological cascades that tells which part of those gigabytes of code to execute, right? There's methylation patterns, there's whole layers of metadata on top of that data that tells which parts of the code to execute. And what that means is that we have biological cascades that are being studied by developmental biologists worldwide to help us understand how do those stem cells become muscle, become neuron, become skin, become bone, in conjunction with each other to, of course, become this ecosystem that we all are. And as an engineer, okay, developmental biology is really interesting, but for us, in our field, it really becomes more like a playbook. What can we learn from developmental biology to help us implement these pathways in the laboratory, right? So what that means for us as a field of tissue engineering is that we have a paradigm called the biomimetic paradigm. So like mimes, right? So biology, mime, copying nature, right? So what this means is we try and copy nature at every step of the process of growing a cell so that at the end of the process, we'll have a tissue that mimics what we have in the body. So we take cells, we put them on biomaterials, we put them in bioreactors, which is basically a word for like a fancy fish tank, meaning, you know, freshwater fish need fresh water and saltwater fish need salt water. What does a bone cell need? What does a heart cell need? How do we copy that in the laboratory using you know, control systems, using digital logic, using all the kinds of programmable, controllable systems, sensors, and actuators that we have at our disposal in order at the end of that journey to engineer a heart cell, a bone cell, and so on. And of course, the cells are the ones doing all the work, but we're designing the environment, okay? And with this approach, I hope to convince you that way beyond medicine, this has application. But first, living implants, right? So our first goal as tissue engineers is to create a one-stop body shop for human organs. And as a field, we've made some impressive feats. This is from Tony Atala's lab at Wake Forest University, where they've engineered human bladders. There's now a person living with a human bladder that's been engineered since 2002, I believe. Don't quote me on that, but over 10 years. From Harold Ott's lab at Harvard University, they've engineered lungs and kidney. From Laura Nicholson's lab, they've actually started a company called Humacyte to produce engineered human vasculature. And in terms of diseases, there are labs that are producing disease models like these stem cells that exhibit properties of Lou Gehrig's disease that can help us understand diseases and cures. And retinitis pigmentosa, which is obviously a disease that matters a lot to my family. And three-dimensional printing of cancer cells in a dish from Karen Berg's lab at Clemson University. Okay, so it's probably not a far-fetched conjecture for this audience to think, of course, this is biology, it has wide application in medicine. But what I really want to convince you is that there are very few industries, I predict, I've been given permission to be a bit provocative here, that will be affected by this type of thinking. This idea of, can I do fill in the blank with cells? Okay? What can be grown, not built? What can be grown, not fabricated, right? And so I'm going to give you a slice of some of the people in maybe the oddest of places that are taking this approach. This is Suzanne Lee. She's a fashion designer and she's the chief creative officer at Modern Meadow that's using this approach to engineer leather in the laboratory. And they've been using this for everything from shoes to leather jackets. Here's Mitch Joachim. He's the co-author on a book that we wrote together called Supercells, and he's an architecture professor based in Brooklyn who asked, why are we building buildings when we can grow buildings using trees and scaffolds like living lattices? It's a very similar approach to what we're using in tissue engineering, but on the architectural scale. This is Damien Palin. He's a biomineralogist based in Singapore, and he collaborates with bacteria to mine the toxic byproducts out of desalination brine. This is creating wealth from waste while protecting the environment. These are the things that we can think about doing if we start collaborating with living systems. This is Ingmar Rydell Cruz. He's a bioengineering professor based out of Stanford, and he realized many people realize that cells respond to electrical signals, but what he did was he employed this in service of creating living video games. Here's an example of a classic video game breakout that was used to inspire this game called Micro Bash. The player guides Paramecia to clear blocks with the ball. Love the 80s aesthetic here, by the way. It reminds me of my bad hair days. And at the other end of the product's life cycle, this is J. Rim Lee. She's developed a strain of mushroom that she calls the infinity mushroom that she's incorporated into a living bodysuit that's kind of like a living coffin that remediates the toxic byproducts released by the body after death. Colbert Report had a field day when she went to the Funeral Directors Guild conference. I don't know if that was held in Vegas, but you can imagine. And this is Sangeeta Bhatia. She's based at MIT, and she's developed a strain of bacteria that glows green in response to the presence of colon cancer cells. This is cancer-detecting yogurt on the horizon, okay? It almost begs the question, what can't be done? And, if these technologies became mainstream, what would they mean for us as a society? And so in the spirit of the theme of this session, I thought I might bring up this quote from Anton Chekhov. The role of the artist is to ask questions, not to answer them. And I love that the field of art is infiltrating into this field of tissue engineering. This is from a lab called Symbiotica based in Perth, Australia, led by two people, Yonat Zor and Oren Katz, who saw this picture of the mouse with the ear on its back and decided as a result to enroll as research fellows at Harvard in order to study these techniques for the purposes of creating bioart. Okay, bioart. And they're very ironic. They do things like ask questions. If pigs could fly, what would their wings look like? And then they took pig cells and they grew pig wings. Okay, they also grew them in green and blue, by the way. They also engineered, have you seen the little bulb there at the top? A victimless leather jacket. This was displayed at the New York City's Museum of Modern Art. And in a science, their lab is called Symbiotica. In a Symbiotica curated show, artist Andre Grodick created an exhibit called Portraits, painted with genetically engineered glowing bacteria. Can you see a little portrait in that petri dish there? So these bacteria were reprogrammed with the gene for red fluorescent protein. This is where we are, guys. We can take genetic codes and we can splice them into living organisms and use them as factories to create everything from art to medicine, okay? And at the same time that this artist transfected, that's the word, overwrote, okay, transfected these bacteria to glow red, he also transfected them with a gene that's been implicated in human Alzheimer's disease. So as these portraits fade over time, they evoke this idea of lost memories in a way that you could argue very few pieces of art really could. So what's next? I hope I'm convincing you that by learning to speak the language of cells, we can coax them into doing a lot. Saving lives, replacing bones, conserving the planet, inspiring unprecedented works of art, providing joy, and hopefully continuing to provide wonder. But that also raises important questions that need to be asked. You know, if we can cut and paste like we can now with CRISPR technologies to the genome, what does that mean for us as a society? Sure, we can think about things like DNA storage, or like 2 weeks ago it was announced at MIT that a programming language for living bacteria. But there are important questions that need to be addressed. Many people say, are we playing God? I think if we unpack that question, we're really asking some deeper questions. Is tinkering with the cellular material of viruses, bacteria, plants, or animals, or humans equivalent? Should there be different bars for tinkering for the sake of research versus education or entertainment, in the case of biotic video games? And if there are more bacterial cells than human cells in our own bodies, and if human cells can be grown outside the body and reimplanted into the body, and our bodies don't seem to know the difference, what does it even mean to say that we're human? What are the boundaries of the human body? These are difficult questions, and they're questions that must be answered, that will be answered. And just in case our blood pressure is going up from our highly responsive cardiovascular systems that sense our stress levels, maybe we can take comfort in the idea that looking back, these questions echo other disruptive technologies that we've encountered before. With the invention of the telegraph, aka the first social media, okay? Back in the telegraph, one of my favorite books is the book called The Victorian Internet, okay, where we discovered that things like online dating and online shopping could exist, right? And when we think about these boundaries of the human body, these questions that arise, we're reminded of in vitro fertilization debates and blood donation. And we got through those technologies, and we figured it out. And so what I would say is that as we think creatively about the future, I think what we need most is collaboration. My theory is that once you see DNA, this is strawberry DNA in this girl's hands. Has anyone ever isolated DNA from strawberries? A few of you. If I get invited back next year, we'll be making DNA cocktails. This is the genetic code that tells strawberries to be DNA, and it takes up an appreciable amount of the strawberry. Every time you're eating a strawberry, you're eating the code that tells the strawberry to be a strawberry. Is that meta or what? Okay, and so what I found is that by isolating DNA from strawberries, we get a chance to appreciate this. And once we see DNA and hold it in our hands, I think we get renewed appreciation for all those data centers that we all work so hard to maintain, or the cloud, right? Data takes up physical space. At our company, Epibone, we try and, even though we're only 8 employees, we try and live this idea firsthand by incorporating an artist in our residence. So this is Maya, our first artist in residence who made bone jewelry based off of the CT scans of the animals we were treating. My idea is that, artists look at, while we as scientists and engineers look for. And if we're going to think creatively about how to digest these disruptive technologies that are upon us, things like DNA storage and fuzziness about the boundaries of our own bodies, DNA patenting, genetic patenting, I think it behooves us to bring people from all of the reaches of our society into the debate. So my theory is that once we see and hold DNA in our own hands, things like crime scene shows will never be the same. So with that, I just want to thank you so much for your time and invite you to ask your most difficult questions, because I think what this really is about is fostering a conversation. Thank you so much for your time and for your invitation.